This video abstract is AI generated. Why is enzyme 1 inactive as a monomer? In this work, we studied enzyme 1, the first enzyme in the bacterial phosphotransferase system. Enzyme 1 starts the pathway by using phosphoenolpyruvate, or PEP, to initiate phosphoryl transfer, ultimately linking carbon uptake to broader bacterial physiology. A key feature of enzyme 1 regulation is oligomerization. The enzyme functions as a dimer, but under physiological conditions it exists in a monomer-dimer equilibrium. The dimer is active, the monomer is inactive, but the structure of the monomer had remained elusive, because enzyme 1 strongly favors the dimer at the high concentrations needed for conventional structural biology. To solve this problem, we engineered a pressure-sensitive version of the EIC domain, called 3MEIC. 3 charged residues at the dimer interface were mutated to weaken intersubunit salt bridge interactions. Importantly, at atmospheric pressure, this engineered protein still formed an active dimer, bound PEP, and hydrolyzed PEP similarly to the wild-type domain. The key step was to combine this engineered variant with high-pressure NMR. As pressure increased, 3MEIC shifted from the dimeric state to a stable monomeric state, while wild-type EIC remained mostly dimeric. Relaxation measurements confirmed that 3MEIC at high pressure behaves like a monomer in solution. We then used residual dipolar couplings together with accelerated molecular dynamics to build a structural ensemble of the monomeric state. The result was striking. Monomerization does not globally unfold EIC. Instead, the overall fold remains intact, but the catalytic region becomes locally disordered. The main structural changes occur in 3 catalytic loops near the dimerization interface: alpha 3 beta 3, alpha 6 beta 6, and alpha 7 beta 7. These loops are ordered in the dimer, but become flexible and partially disordered in the monomer. This local disorder has a direct functional consequence. NMR binding experiments show that monomeric EIC does not bind PEP, and activity assays show that it cannot hydrolyze PEP. Without dimerization, the catalytic loops cannot organize the active site for productive substrate binding. Together, these results explain why enzyme 1 requires dimerization for activity. Dimer formation orders the catalytic loops, creates the PEP binding site, and enables phosphoryl transfer. The inactive monomer is not unfolded, it is locally disordered at exactly the regions needed for catalysis. To know more, read our work online.